Hello and welcome everyone. In this session, today we discuss about leaf curl of tomato. The disease has been reported from India, Sri Lanka, Nyasaland and South Africa. In India, when the plants are infected within 20 days of planting, or the loss may be up to 92%, while infection of 35 and 50 days. Old crop result in 74 and 29% loss, respectively. Shastri and Singh, 1973. Symptoms: the leaf curl is characterized by severe stunting of the plants with downward rolling and twinkling of the leaves. The newly formed leaves show chlorosis. The older curled leaves become leathery and brittle. Stunting of the plant is due to shortening of the internode. The diseased plants look pale and produce more lateral branches, resulting in a bushy growth. There is partial or complete straightening of the plant as associations of leaf curl with root node increase damage to the tomato plant. The causal agent: leaf curl of tomato is caused by the tobacco leaf curl virus, Gemini virus growth. The same virus causes leaf curl of papaya and leaf curl of chili. The virus was first noticed in 1912. Its transmission was described in 1931, and the virus was characterized in 1978. Its synonyms are tobacco capping virus, tobacco curl leaf virus, tobacco trenching virus, tobacco leaf curl virus, first tobacco virus 16, and nicotians virus 10. The particles of the virus have germinate structure, that is, they are made up of two incomplete icosaedra, icohedra, isometric practices particles with one shared subunit and measure 15 to 20 by 25 to 30 nanometer. The nucleic acid is a circular DNA molecule. No information is available on its biological properties in SAP, such as thermal inactivation point, dilution and point, and longevity in vitro. Although it is a Gemini virus, it has no serological relationship with any other member of the group. The virus is present in phylum where it causes phylum proliferation in stem apis, petioles, and ventlets and veins of leaflet. Particles occurs in closely packaged areas, forming ridge road in some of the nuclei of phylum cell. These rods are 25 nanometer wide and consist of row of germinate particles. Nucleus is split into granular, and fibrillar regions. In addition, tomato, tobacco, papaya, and chili, other natural hosts of the virus are Agranthum conjundius, Euphaetum odoratum, Euphorbia hirta, Solanum nigrum, and Vernonia cinerea. The virus can infect Datura stramonium. A number of isolates differing in a host range or severity of symptoms produced in tobacco have been reported. Most isolates contain a mixture of strains of variants. Some isolates lose infectivity on tomato after passage through tobacco and vice versa. The virus is neither seed nor sap transmissible, but external contamination of seed may occur. Daughter transmission has been reported in India. The main agency of its transmission is the whitefly Bemisia tabaci, and vector can transmit the virus after a 20-minute acquisition access period of 4 hours. In India, the virus is reported to be transmitted by the vector after a 30-minute acquisition access period of 6 hours. The time required for symptom expression varies, varies in different cultivar of tomato. Whitefly population per plant also varies with host cultivars. Even a single viruliferious whitefly is able to transmit the virus. The virus is persistent in the body of the vector and is retained for more than 12 days or for the whole life, but there is no evidence that it multiplies in the body of the vector. There is no transfer of the virus to the progeny by the vector. Reddy and Yaragun, Tia, 1981. Management of the disease. No highly effective control measure against leaf curl are known. Lycopersicon parvinum, a species related to cultivated tomato, is highly resistant to leaf curl. So far, this resistance has not been transferred to commercial varieties. Some workers have claimed good control of the disease through reducing reduction in the population of the insect vector. Use of systemic insecticides such as methyl parathion 0.02% and dimethoate 0.05% as a spray of carbofuron furadone at the rate of 1.5 kg per hectare. As soil application have been found useful in preventing build up of vector population and thus reducing the disease severity. Spraying should be started soon after planting. 5 to 6 sprays are required. Carbofuran, soil treatment of nursery bed, beds and the man field not only reduces the disease by about 70%, but also increase the yield manifold. There is also a reported that increasing the density of plant population from 20 to 90 plants per square meter and three applications of parathion 0.02%, dimethionammethyl 0.02%, or dimethoate 0.05% reduce disease incidence. A single application of folate thymate at the rate of 50 kg per hectare or carbofuran furadone at the rate of 40 kg per hectare at planting time is also highly effective. The treatment protects the plant against whitefly attack at least 40 days. There are experimental evidence that growth regulators like gibberellic acid and 2,4-D reduce incidence of the disease and greatly increase fruit yield. Hayati and Verma 1985 have reported that foliar application of the antibiotics validomycin A 0.02%, avomycin A 0.01%, and of guanidine azoguanidine EPC suppresses the disease. So thank you everyone to listen me and stay connected.